Good day everyone. We are students from Far Eastern University, Manila, taking up BS Biology, major in Medical Biology. We are going to show you the process of collection of specimens itself, to proper mounting and to the identification of the specimens, specifically of the family Formicidae or ant in common name. We hope you are going to enjoy this video while learning some of the process and the key importance of this video relating to medical entomology. Medical entomology is the branch of science that deals with the study of insects that cause disease and their medical importance. It is an important subject as it contributes to the everyday lives of humankind. It detects the role of insects in ways of discovering the diseases they carry, their role in the environment, and even protection from the disease, and also the natural flora. Throughout the next series of activities, we will show you the vital steps in understanding the importance of the insects and the subject by first collecting a sample, processing the collected sample, mounting the specimen, and the last step is identifying and observing the specimen. This activity will intend to better understand the biology of insects by identifying the taxonomic key of the specimens, including its class, order, family, subfamily, genus, and species. Second is to be familiarized with the external morphology and key characteristics of the specimen, as well as bearing the importance of the processes of collecting the samples up to identifying a specimen.
The equipment used to assemble a general insect or mite collection need not to be elaborate or expensive. In many instances, a collecting net or several killing jar or bottles will suffice. However, additional items will permit more efficient or effective sampling for a particular fauna. Many collectors carry a bag or wear a vest in which they store their equipments that they need. So the following items are the basic materials used for the insect collection. We will use a 1/4 sheet of bond paper where the baits such as the peanut butter and tuna will be placed. The insects captured through passive collection will be stored inside the ziploc along with the 1/4 sheet of bond paper. So for our baits, we use the peanut butter and the tuna, and these are the good choices that will cater all the nutrients or diets of different insects that we can gather. And these can be placed on the 1/4 sheet of bond paper. Alcohol aids in insect collection as it stuns the insect for easier collection. For increased efficiency, it is recommended to use the sprayer. The insect net is very useful in catching insects that fly, such as those that belongs to the order of Lepidoptera. The killing jar is effective in collection of insects and related groups that is usually requires that the specimen be killed so that they are properly mounted or to be studied. Any heavy, wide-mounted glass jar or bottle with the tight-fitting stopper or metal screw top may be used. The killing agent used may be any various liquids or solids. Liquid killing agents generally are considered to be slower acting but safer to be used than the solids such as cyanide. In order to achieve a complete set-off, the following materials are needed. The insect specimen should be prepared prior to the actual mounting process. Entomological pins are very useful and very important for entomologists for mounting the insects. Another material that is important is the entomological puncture. It is used to create the paper tips where small specimens such as ants will be placed. We also use the pinning block. It is used to pin the insects of various sizes simultaneously. The block therefore negates damage caused by pushing the mounted specimen into the flexible surface such as the styrofoam. A pinning block has a series of holes of different depths drilled in it to mount specimens and label at standard height. We also use the water-based glue, and it should be used to stick the specimen to the paper tips. We also use a tennis ball and we cut it into two, and then we filled it with a cement. So it is also referred to as an iguchi ball, is where the entomological pins will be placed whilst the actual mounting process. So we also use a probes, and another alternative way to create the probes, we use the sticks and also the pins, and we tied it up, and it is used to assist in adjusting the insect on the paper tip. So it is very necessary to label every specimens that are mounted in order to avoid confusion in identification between the specimens that you gathered.
There were three collection methods used for this activity. First is with the help of sweeping net for collection of insects. The vegetation around the nursing building was swept with the net, and the larger insects collected is placed inside a killing jar, with ethanol as the killing agent. The second method is the active collection, which is hand-picking. In hand-picking, a test tube that contains ethanol was prepared. The insects of interest are stunned using alcohol and then are picked up using a tweezers. The insects caught are then placed in the pre-prepared test tubes to kill them. And the last collection method that is used is the passive collection, which utilizes baits. The peanut butter and the tuna bait is placed on a paper, then placed in the first floor of the nursing building in areas near possible location of the lair of the insects. The bait is allowed to sit for 15 minutes until insects are seen pulling the area. They are then sprayed with alcohol, rendering them stunned. They are then put into a Ziploc bag. All the insects collected were present in the vicinity of nursing building of Far Eastern University.
After collecting the specimens, we proceeded on mounting our collected insects. We set aside the collected specimen on a tissue paper to absorb the used preservative before mounting, which is ethanol. After that, we used the specialized point punch or the entomological puncture to create enough paper tips. We also made sure to have extra pieces for future use. The paper tips are then carefully attached to the entomological pin, then with the pinning block, which is used to set the paper tips to the desired height. After that, we put the pin on the Iguchi ball to proceed to the next step. Then, the next thing that we did was to focus the paper tip under the dissecting microscope. It is for us to properly place a small amount of water-based glue to the very tip of the paper tip using the entomological probes. After that, with the help of the probes, we made sure that the specimens are properly mounted on the tip of the paper stage in a lateral manner with its head on the left, and the tip of the paper stage is in between the first and second leg of the specimen. And lastly, with the help of the entomological keys by General and Alpert 2012, provided by our professor, we identified the specimens that we have collected. All of these specimens were stored inside the tackle box cushioned with styrofoam. Each mounted specimen were given data and determination label indicating the location, the date, and the name of the collector of the specimen, as well as their order, family, subfamily, genus, and species.
System classification helps us to bring order to a difficult environment. Organisms in biology can be classified based on their overall resemblance or based on their evolutionary relationships. When it comes to categorizing species, most current scientists use a cladistic method. In biology, organisms are assigned a generic name and a specific name. A genus is a collection of organisms that are closely related. Closely related genera are placed into a higher, less specific category known as family. Families are grouped as orders, while orders are grouped into classes. Classes of the organisms are grouped as phyla, and phyla are grouped as kingdoms. Lastly, domains are the organism's highest taxonomic rank. In order to further identify an organism's domain down to its species, dichotomous keys are used, which is a method of identification in which groups of organisms are repeatedly split into two categories. First is with each successive division, additional information about a certain organism's characteristics are revealed. Second, it has been identified when the organism no longer shares all of its chosen with any other organisms. It is recommended to utilize immutable traits when using dichotomous keys to identify specimens. The group gathered ant specimens for further identification. Based on observation, it is concluded that the subfamily of the ant-1 specimen are Myrmicinae, due to its distinct features: antennal sockets partially and completely concealed and having a presence of postpetiole. Whereas the subfamily of ant-2 specimen are Formicinae due to the sting being absent and the notable feature being the presence of acidopore that is frigid with hairs and the absence of a postpetiole. In conclusion to this activity, the group performed different techniques such as collection, processing, mounting, and identification of the specimen. This activity gave more knowledge to the students about entomology. The group were able to identify the taxonomic keys of the different species and also the different parts of their bodies. This real-life application of the different techniques in entomology made the members to better understand what entomologists do. Also, this activity helped the members in identifying the different subfamilies, genus, and species of ants. Other than that, the members also learned to identify the morphology and the key characteristics of every specimen they've mounted.